presenting you my basically my PhD research. So thanks to the Society of Evolution, Study of Evolution, and Rosemary Grants for supporting my research, and also this research was done under the supervision of Dr. Carlos Prada at the University of Rhode Island. So, as already introduced, I'm going to be talking a little bit about the speciation and also heat tolerance and bleaching on reef building corals in the Caribbean mostly, or specifically in the Caribbean. So, as probably many of you already know, coral reefs are among the most biodiverse ecosystems on the planet. They are estimated to be home to around 25% of all marine species known to science, with many more species relying on coral reefs to complete part of their life cycles. But they're also important in terms of ecological services to humans, in terms of coastal protection, food security, cultural heritage, and they are also a big reservoir of unknown chemical compounds that could be relevant for future research in terms of biomedicine. But one of the things that makes coral reefs truly unique is the fact that reef building corals, stony corals, the organisms responsible for engineering coral reefs, are able to harvest sunlight. And through this ability, they are able to construct structures such as coral reefs that sometimes can be so large they can be seen from outer space, like as we see here on this reef in the Caribbean, from a picture from the NASA. But behind these massive structures, what underpins everything is the symbiotic relationship that reef building corals have with microsymbiotic algae. These photosymbiotic algae allow corals to actually harvest the sunlight, while the corals provide a safe environment for the algae to proliferate and live happily, we could say that. But something that we know during the last decades is that this close symbiotic relationship that underpins the formation of coral reefs can be easily disturbed by thermal stress. So as temperature increases on oceans, and mostly because of since the onset of the Industrial Revolution, because of all the OC that I was putting out in the atmosphere, this close relationship can be easily disrupted, and corals either expel their microsymbiotic algae or they start to digest that algae, and therefore that creates that pale or white color that we know as bleaching. This bleaching is being responsible, while it's not the only single cause of coral cover decline across the world, is one of those major causes of massive die-offs of coral reefs across tropical environments. So as we can see here, we've been having a decline of coral life cover, and obviously this is also true to many ecosystems as well, right? We're losing our biodiversity as we speak. But something that is being known for several years now to coral reef researchers and biologists is that the susceptibility to bleaching, even though all corals are pretty much susceptible to bleaching, there is a lot of variability, not only between species or among species, but also within individuals within a species. So basically, as we can see here in these pictures, we can have here on the left or my left a coral reef community where different species have different bleach phenotypes or bleach individuals, but also within what we consider single species, we can have individuals that happening in the same reef, like no obvious differences between species or environments or morphology. They can have different bleaching susceptibilities. Therefore, some colonies bleach, some others don't, or the degree to which they bleach is different. And we also known for several years, similar to many other species, is that there are more species that actually meet the eye. So species that sometimes look similar can be different, and sometimes species that don't look alike can be considered the same species. So we know that the diversity of coral reefs or coral species is more than we have actually described. In a recent review, I participated with some wonderful collaborators at the Boston University. We review extensively the literature documenting divergence within sympatric lineages of reef-building corals and also soft corals across the globe. And just as a reminder of what a cryptic species is, we can have just two species that look exactly the same. Here there is the different color, just to illustrate that they're different, but morphologically they look very, very similar. And it's not until we start doing, and we never know where along the continuum of speciation these species can be. So it's only until we start gathering molecular, morphological, and other reproductive biology data that we can start like teasing a species out, you know. So we found that there are multiple reports of coral cryptic lineages. But one of the trends that emerged from this review, and this has been known for several years, is that these coral species can have like distributions across environmental gradients, such as distance from shore or the depth at which you find these cryptic species. But also they can have variable symbiotic relationships, meaning that sometimes one species or one lineage can have very high fidelity in terms of the symbionts that they have, or sometimes that can be very variable, where different lineages can have multiple symbiotic relationships, or just one lineage can have multiple symbionts. So there is a lot of variability here, and we're still kind of getting our heads around what is going on in terms of symbiotic relationships, cryptic lineages, and environmental variability on coral reefs. So within that frame of cryptic lineages, species biodiversity on coral reefs, and thermal stress and bleaching is the kind of working frame within which I establish my research questions. So the first question that I ask is whether closely related coral lineages differ in their physiology, because physiology underpins how they respond to environmental variability. Mm-hmm. And also, I ask whether those physiological differences actually matter for coral bleaching. That is, whether differences in physiology can have different susceptibility to bleaching. And finally, gearing more towards evolutionary biology, I study how coral or lineages can actually diverge and the evolution of the divergence of coral lineages. So with that, let me introduce you to my study system. Being studying Orbicella species complex in the Caribbean. These are stony reef-building corals, which are major reef-building corals in the Caribbean and Gulf of Mexico. These are composed of, this complex is composed of 3 species, Orbicella fabulata, Orbicella annularis, and Orbicella franksi or franksi. They are sister taxa that have evolved in the last 500,000 years or so. And here we can see like a phylogenetic tree of the relationship between these species complex. I'm going to be talking about mostly about Orbicella fabulata and to some degree Orbicella franksi. So in terms of one of those major structuring factors on coral reefs, it is depth, and that is also true for this species complex. We have that overall Orbicella annularis is normally found in shallow waters. Orbicella fabulata, which is the species that I studied the most, has a broader depth range, and Orbicella franksi is typically restricted. I think there is. I think someone is not unmuted, so if you could just please check that your all guys are muted, I would appreciate it. Thank you. So I was saying that Orbicella franksi is typically found in deeper water. So we have a depth segregation, and that depth segregation is also related to how they can use the light available at different depths. But also in the very beginning of my research, I found that there is evidence of cryptic lineages, that is diversity within what it was thought to be just one species, but actually we found that there are more than at least genetic clusters, if you wish, within one single species. And this was published in Molecular Ecology. So to give you like an overview of what we did here is that originally we reanalyzed a study that had used Orbicella fabulata, and they used thermal stress to quantify bleaching in Orbicella fabulata in Panama, in Bocas del Toro. However, we found that certain patterns of genetic variation, specifically transcriptomic variation, probably were more related to variation within multiple clusters of genotypes and not so much within a sympatric group. So what we did is that we reanalyzed that data, and based on around 3,000 single nucleotide polymorphisms, we found that what the researchers originally thought was just like variation within just one single species actually conform more to different, at least, lineages or different independent evolutionary units here found in this discriminant analysis, component of discriminant analysis, and also using phylogenetic approaches. So we found that the variation in terms of bleaching could be related not so much within intraspecific, but actually more interspecific variability. So the way the researchers quantified this was they subjected the colonies to thermal stress and then quantified the degree in which they lose their symbiont. So in a negative change meant that they bleach more, and a positive change here meant that they didn't bleach much. So as you can see here, when we look at the association between lineage and also the number of colonies that belong to that lineage and bleach, we can see at least a weak relationship in terms of like the red lineage was the one that was more heat tolerant. So that was kind of my starting point in order for me to study cryptic lineages and variation and physiological variation within this species complex. So now back to my research questions. The first one was, do closely related coral lineages different physiology? So for this, because we know that one of the major structuring factors also associated with cryptic variation within coral reef lineages is depth, we went to Media Lunar Reef or Half Moon Reef in Puerto Rico, where we know that there is a marked decrease in light availability just within around 20 meters or 30 meters. Therefore, colonies that are in the shallow areas are exposed to a lot of light, and colonies that are even not so deep as 20 meters can see very little light compared to the shallow area. So in terms of our experimental approach and how to study this, we conducted genome scheming using low coverage whole genome sequencing. We also identified the symbiont by studying the internal transcribed spacer for corals, for the symbiont, in corals. We also quantify physiological measurements. We look at chlorophyll A, protein content, the density of symbionts, and also we characterize the photobiology of this coral and symbionts. Further, we conduct reciprocal transplants to test for local adaptation and also to see how differences affect in the different genotypes across depths. And finally, we use acute thermal stress assays to actually see the effect of thermal stress and how that affected the bleaching phenotype. So the first thing that we found is that using genome scheming for around 3,000 single nucleotide polymorphisms, we clearly could distinguish Orbicella franksii and Orbicella fabulata. As you can see, two different clusters here on this PCA. However, not so much difference between Orbicella fabulata across shallow and deep environments. But as we progress on the bioinformatic analysis of this, we could at least see that to some degree there is some genetic differentiation across environments within Orbicella fabulata. Then we look at the symbiont associations for each species at each depth. And as you can see here, Orbicella fabulata in the shallow environment was dominated by the genus Symbiodinium in blue, although some colonies also have some other symbiotic associations. And again, within the same species, but at a deeper depth, we see that symbiotic association or community actually changes, where we start seeing more variability in the composition of those symbionts within the colonies. And then finally, for Orbicella franksii in the deep environment, because remember that Orbicella franksii is typically restricted to the deep environment, whereas Orbicella fabulata can occupy a broader depth range, we see that variability resembles more like the one found in Orbicella fabulata, but also we found here a pattern in which there seems to be high fidelity in the sense that each individual colony of this species actually only had one of the symbiont. After that, we conducted reciprocal transplants, moving colonies from the shallow environment back to the shallow as control, and then from the shallow to the deep, and the other way for the deep ones, deep to deep as control and deep to shallow. And we leverage the microfragmentation method, which is one that, taking advantage of the clonal nature of corals, from the same genotype, we created multiple little fragments so that we could have replicability across genotypes, and therefore we could have multiple individuals. You could call individuals because they were just the same genotype but just smaller fragments for each one of these reciprocal transplants, and we transplanted over 2,400 fracs. So in terms of the physiological analysis that we conducted, we look at the protein content, the symbiont density, the levels of pigment or chlorophyll A, the photosynthetic efficiency, or how well they can utilize the available light, and also the metabolic demand in terms of respiration. So in order not to walk you through all these measurements, I'm just going to give you an example of respiration so that you can see what we found. And also in terms of, for doing physiological analysis, we also had like a smaller data set, and it was very, very intense to do this work. And this was done in collaboration with the Iglesias Prieto lab. And I'm actually not myself a physiologist, so I do thank here Tomás González and Naomi in Iglesias Prieto lab for helping me do this analysis. So here what we have is Orbicella fabulata and Orbicella franksii. We have the native shallow environment, the native deep environment, and the native deep environment for franksii. And here we have the native transplants, shallow to shallow, deep to deep for native environments, deep to deep for the species. And then we have the reciprocal transplant, shallow to deep to shallow, and deep to shallow. So as you can see, when we look at the shallow for Orbicella fabulata and deep, we can see, and then compare it to franksii, we can see like an overall trend of decline in terms of respiration, meaning that their demands of oxygen vary across species and depth. And we see actually the response of when those colonies from the native environment were transplanted into the foreign environment. So this was one of the trends that we were able to found. So in order to characterize this overall variability in physiological measurements that we took, we were able to kind of categorize each species and their habitat depending on how much variability we found in terms of all these physiological measurements that we took. So we categorized or broke it down into 3 categories: high, intermediate, and low metabolic demand. So as you can see, Orbicella fabulata in the shallow environment has a high metabolic demand, has a lot of protein, symbiont density, little chlorophyll A, has a high photosynthetic efficiency, and a high respiration demand. For the same species in the deep environment, we found more like a sort of intermediate. And for Orbicella franksii, we found that is the one that has like the lower physiological demand, or the lowest metabolism. So, okay, we found genomic differences to some degree within a species and also between species, variability across environments, but how does that matter for actual differences in survivorship in terms of reciprocal transplants? So we monitored survivorship across 6 months, and for instance, for Orbicella fabulata in the shallow environment, that let me remind you that it had a high metabolic demand. When we moved colonies from the shallow back into the shallow, we found around 75% of survivorship after 6 months. However, when those same individuals that were originally found in the shallow environment were moved down to the deep environment, we found a marked decrease in that survivorship, well below 50%. So we can see that they don't do well when they are moving to environments with low light. However, when we look at the data, this time for Orbicella fabulata coming from the deep, you can see that in the native environment, the survivorship is over 80%. And remarkably, in the shallow environment, they'll do as well as they do in the native environment, meaning that if you move colonies into the shallow environments from the deep environment for these species, they don't seem to care much about that light, or they survive very well. And similarly, we found that the same was true for Orbicella franksii. Remember that only occurs in the deep. So in the deep, it had around 85% of survivorship, and in the shallow environment as well. So what we concluded from these reciprocal transplants was that colonies that are used to seeing a lot of light, like Orbicella fabulata shallow, they don't perform well when they are deprived of that light. However, when you move colonies that are acclimated to a low light environment into a well-lit environment, they do much better. And this has repercussions because we've been trying to restore coral reefs, and sometimes those restoration efforts don't take into account environmental variability and particular susceptibility of colonies or species to environmental variability. And unfortunately for us, for most of the restoration efforts, colonies over time, they die. And very little success we have had in restoring coral reefs. So it was more difficult than we originally thought, unfortunately, for coral reef researchers, and unfortunately for the benefits that coral reefs provide to us. So takeaway so far in this for this part of my research, we found that closely related species differ genetically, but also physiologically across depths, and that the environments they come from actually matter when we reciprocally transplanted them. That meaning that high light acclimated corals do poorly in dim lit environments, but the opposite is true. When we have colonies from the low light acclimated environment, they tend to do much better in shallow environments. So it's important to understand how coral species and particular habitat conditions and physiological conditions determine environmental change or responses to environmental change. Now moving on to my second questions, because I just found you that there are differences in terms of genetics, in terms of symbiotic associations, and also in terms of survivorship. How does this affect actual response to thermal stress and the bleaching phenotype? So in order for us to test this, we use again colonies from fracs from the reciprocal transplants. And what we did this time is that we subjected those colonies to thermal stress through the coral bleaching automated stress system, which is an acute thermal stress assay where we have one control tank, which was the environment temperature. And we had also 2 experimental tanks. And in each one of these tanks, we placed fracs that came from the same genotype after they have been in the field for around 4 to 6 weeks. And we measured the bleaching phenotype through pulse amplitude modulation, which is basically looking at the integrity of the reaction centers of the chloroplast in the symbionts. So whenever this is disrupted, that pulse amplitude modulation is going to change, and that's going to give us a proxy for how much symbionts and how light is being utilized by the remaining reaction centers in the cells of the chloroplasts. So to walk you over the experiment, we collected the colonies and acclimated them for around 18 hours. We measured PAM, and then the experimental tanks were subjected to an acute thermal stress for a duration of around 8 hours. The increase in temperature here was actually 6 Celsius degrees. And then after an overnight rest for the corals, we measured again that bleaching phenotype using pulse amplitude modulation, or PAM. So what we found here is that overall there was a decline in photosynthesis or photosynthetic efficiency. What you're looking at here is each one of the environments, the species, environments, and the reciprocal transplants: shallow to shallow, shallow to deep, deep to deep, deep to shallow, and so on. And in blue, we have the ones at the environment temperature, 28.5, compared to the ones at 34.5 Celsius degrees. And as you can see, there are significant decreases in the in the FB/FM, which is like the proxy that we use for measuring how well they were photosynthesizing or using light energy. So for all of them except for Orbicella franci in the deep for the native transplant, deep to deep, is the only one where we didn't find significant differences in overall performance, even though some individuals here, as you can see, pretty much were almost, they lost most of the symbiotic algae because the decline in photosynthesis was pretty remarkable. This could be also attributed to some individuals that probably didn't start the experiment very well in terms of the photosymbionts that they have, but still I think we had enough individuals to make sure that we could compare this statistically. And in fact, we found that Orbicella franci from the deep environment, even though had individuals that decreased dramatically in that photosynthetic performance, was the one that was more heat tolerant. However, when those same genotypes were exposed to a shallow environment, this is deep to shallow, we see that the heat tolerance was lost. Therefore, meaning that even though Orbicella franci survived well in our typical transplants, they did not respond very well to thermal stress. So even if individuals, or even if one species responds well to a new environment, that doesn't mean it's going to respond very well to when another factor changes in that environment. And therefore, we could conclude that the heat tolerance that we originally found in Orbicella franci, was lost when it was moved to a shallow environment. So with that, I've shown you that there are differences between species in their heat tolerance. We found that Orbicella franci was more heat tolerant than Orbicella fabulata overall, but that heat tolerance actually decreased or was lost when they were coming from reciprocal transplants at shallow environments where there is more light. So here we're looking at the combined effect, and something that I didn't study, but I realized then that is of like high importance, is like understanding the interaction of light and temperature in coral bleaching. And this is something that many other, this is like a new field. We know that also that light affects or has an influence in how corals bleach. So now I'm gonna kind of change gears here, and I'm going to talk more about evolutionary biology within the same species or study system. And one of the questions that I'm really interested in is the evolution of coral lineages, or the evolution of diversity on coral reefs, specifically within coral species. So as I was saying before, we know that coral reef diversity is huge, but we don't understand yet the precise mechanisms responsible for promoting and maintaining that rich diversity on these coral reef ecosystems. And this is so because when it comes to the formation of a species in the marine realm, we have a better understanding of how this happens, typically at larger biogeographical scales, and normally associated with strong geographical barriers, such as the example here of the closure of the Isthmus of Panama that isolated populations from these two ocean basins. And on top of that, for many parts of the ocean, such barriers are not so evident. Plus the fact that many marine organisms have planktonic larvae that are able to disperse several dozens or hundreds of kilometers. So all these aspects combined should, in theory, preclude or at least hinder the formation of species. But as I've shown you, one of those major factors structuring coral biodiversity on coral reefs is depth. And this, of course, doesn't come as a surprise given that the corals rely on light to actually survive, or at least reef-building corals. So we know that depth is one of those major structuring factors in segregating a species and segregating coral reef communities. But it turns out that that light is not only important for energetic budgets in corals, it's actually central to many of their life cycle stages, from inducing or cueing spawning or reproductive behavior, also guiding or relying on light signals for larvae to actually navigate the ocean and found a suitable place where to settle. And of course, the establishment of the photosymbiotic relationship. So light is central to all coral reef biology, or to rebuilding corals biology. We also know that some of the environmental cues that corals rely on to time their reproductive behavior and reproductive effort is the fact that they rely on light signals both from the sun and from the moon. So it's been found that they rely on a combination of signals that if they don't occur in synchrony, or there is a sequence of signals that they rely to actually know that that they can, this is the right time for spawning, if we could say so, is that for most broadcast spawners, they spawn after sunset. So they rely on the sunset signal, the lack of sunlight, a period of darkness, and then the particular wavelength signal from the moonrise. And that's what triggers coral spawning. And also we know that within this system that I'm studying, the Orbicella species complex, the Orbicella annularis and Orbicella franksii, there is a 2-hour difference in the time of which these two species spawn within a single reef. So what you're looking at here is in Bocas del Toro, Panama. This is Orbicella franksii, this is Orbicella annularis, and we have the number of colonies and the time after sunset at which they spawn. So you can see that Orbicella franksii, which is normally found in deeper waters, tends to spawn like around 2 hours after sunset, whereas Orbicella annularis, that is normally found in shallow environments, spawns a little bit later, 2 hours after franksii. So this time difference is what is believed to be responsible for the divergence, or at least the maintenance of the reproductive isolation barrier. This is a temporal reproductive isolation barrier keeping a species apart. Because when we cross gametes in the lab for these species, they are compatible to certain degrees. So my question, back to my central question, is how have divergent coral lineages evolved? So for this, I focus on Orbicella favellata because it's the one species that is found across broader depth range. We look at some morphological variation. We also did some coral genomics across the depth gradient. We conducted outlier loci scans, or look for regions of highly differentiated FSTs, or genomic regions of interest. We look for genes close to those regions, and finally we look at the function of those genomes, or of those genes, that have been characterized for coral reef biology. So again, back to our coral reef in Puerto Rico. Here we collected around 100 colonies from 6 down to almost 20 meters. And this is the number of colonies that we collected at 5 different depths: 6, 9, 12, 18, and 19. And our designation of shallow for 6 and deep for 19 meters, and the number of colonies at each one of these depths. So the first thing that we did was we look at some morphological variability in terms of the polyp density, that is the number of polyps that we found within a certain area. Here we have depth, and here we have, along the x-axis, we have mean polyp density. So what we see here is that in shallow environments, colonies tend to have more polyps, whereas in deeper areas, the polyp density decreases. And this could be attributed to the fact that in areas where there is a lot of light, having more polyps also protects from the excess of UV light, because it creates a self-shading mechanism. Whereas in deeper areas, those polyps need to be more spread out in order to maximize light capture, because there is less light available. Then we conducted genomic analysis using GBS. In this case, we have a smaller data set of around over 10,000 SNPs. But we found that there are 2 distinct genetic clusters for this, at least for this genomic data that we collected. And when we look at the distributions of those colonies, now knowing that they belong to 2 different genomic clusters or lineages, we found that they are segregated by depth. So we have an orange lineage that dominates at 6, 9, and 12 meters, but we start seeing the appearance of the deep lineage, or the one in purple that we called deep, up down to the point where at 19 meters, we only found the deep lineage. And again, if we look back at the morphological variation, but this time knowing that they belong to two different genomic clusters, we see that there seems to be a strong association between the number of polyps and the number and the lineage that they belong to. However, when we look back again at all our variables, depth, lineage, and variability at all of these depths, we see that morphological variation is not so clear at mid-depth, which is something that it is to do with phenotypic plasticity, because we know corals are highly plastic animals. They can respond to the environment in which they are found. But the fact is that just by looking morphologically, at least for the polyp density, which was the only thing that we look at for this case, it's not easy to tell those lineages apart, right? There is a lot of overlap in that metric. So the question that comes to mind is how these lineages originate, because I've shown you that within just one single reef, within what we thought was just one single species, there are two genetic clusters, or lineages. They differ to some degree morphologically, but mostly genomically. So in order to try to make sense of that pattern that we found in terms of genetic segregation, we refer back to what we know about the reproductive biology of related species. So remember that I was telling you about the difference in spawning time of Orbicella franksii and Orbicella annularis. We know that this difference time, this temporal reproductive isolation barrier, it is to do when they see that light, or when they perceive that particular signal that triggers spawning. Orbicella franksii senses that signal much earlier after sunset, the Orbicella annularis, which senses that signal much later. So we believe that a potential mechanism could be acting here, in which shallow colonies of Orbicella fabulata are actually sensing the light much later, or the deep ones are perceiving the signal earlier compared to the shallow ones, because of the decrease in light and the changes also in wavelengths. So in fact, when we compared the genomes of those two lineages by conducting a classic outlier analysis, we found that for those genes, or for those SNPs that were highly differentiated between those the genomes of these two lineages, we found that many of these SNPs were close to genes that have been reported to be involved in controlling reproductive behavior. Some of them belong to GPCR, so G protein coupled receptors, which, as probably many of you already know, are environmental sensors. They are key in how organisms at the cellular level, they can sense the changes in the environment. So knowing that spawning is environmentally regulated in corals, mostly related to sunlight and to moonlight and the combination of these signals, and the fact that we found that there are GPCR genes, particular ones that are involved in rhodopsin-like proteins, because GPCR is a huge family of proteins, but we found some of those genes that are related to this family of rhodopsin-like that are responsible for sensing the environment, particularly light, but also chemical cues. And the fact that so many other studies have characterized many of these GPCRs in sensing and triggering, or involved at least, the down regulation or up regulations of the transcripts of those genes prior and during and after spawning, have been characterizing during coral spawning. But also these GPCR genes are involved in how larvae can sense the environment as they navigate the ocean when they are released. And also that could determine where they settle, because we know that coral larvae can preferentially settle not only on certain substrates, but also the presence of certain algae there. And also sometimes it's been found that the color of the substrate can determine how or where these coral larvae settle. So the interaction of how they sense light and how they sense other chemical cues in the environment when they spawn and also when they settle.
Did you fix the dryer?
Sorry, could you make sure that you're muted? Thank you. So I was saying that the combination of the role of GPCRs um, in sensing light or sensing other environmental cues when they are timing spawning, and also when the larvae are looking for places, sort of looking places where to settle, could create a positive loop of reinforcement on where these individuals can settle and how they overall or when they overall reproduce. Moreover, we also estimate from changes in allele frequencies from our genomic data. We estimated selection coefficients across a very, very narrow linear distance, less than 100 meters across depths. And we found that from Klein theory, we could estimate selection coefficients which were quite strong for these particular genomic regions. And similarly, when we compared some of these coding protein genes across different species, or between species, Franci and Fabulata and Franci and Alvarez and Franci, we found that for some of these areas, there are increased DNDase ratios, meaning that this is like a good way forward to actually explore the role of these genes in how corals can speciate or diverge. And that led us to propose that the evolution of GPCRs in corals could explain patterns of widespread patterns of speciation across depths in corals. What you're looking here is part of the review that I showed before, but also part of the research that I conducted, where I look at the report or those reports or studies reporting lineage divergence within sympatric areas and also within some genus or species of corals. And this is an overall phylogeny of some of the families of corals and where those divergence within lineages have been found. So this could represent the evolution of GPCRs could represent a good avenue for studying coral speciation and segregation across depths. So the takeaway so far, or that to conclude pretty much, because I'm towards the end of I'm basically done with the talk, is that we found that there is genomic divergence across a depth and light gradient that shows some cryptic divergence within coral species. We found that this genomic divergence is related to proteins responsible for sensing the environment, such as rhodopsin-like proteins, and that the evolution of these environmental sensing proteins could be a mechanism through which corals could diverge and adapt across depths. So with that, to conclude, I hope that I've shown you that correctly identifying corals and their particular physiological susceptibilities actually matter for conservation under climate change, particularly under global warming, because, and this is also true for many of the other species or the other systems that you guys study, is that biodiversity is grossly underestimated, and that's true for building corals and for many other coral taxa. And this is important because we need to understand the distribution of those species, where we have species that we thought or that we think are widely distributed, or actually they are most geographically or even within very geographic ranges are more restricted. And also understanding the particular physiological susceptibilities of those individual species or lineages to environmental change, because this overall can help us understand current and future or forecast future ecosystem dynamics. So with that, I'd like to thank my PhD committee, Dr. Carlos Prada, my main advisor, as well as the two other members of my PhD committee, Dr. Andrea Puccini and Graham Forrester, and also acknowledge all the other, what you saw here is not just my research. This is part of a huge collaborative effort among many researchers across different career stages and across different institutions and nationalities. So without them, I wouldn't be here giving you this talk. So with that, I thank you all for your attention and for being here. Gracias a todos por estar acá, and I'm happy to take questions.
All right, everyone. Thank you, Matthias, for a great talk. Really enjoyed all of that work and learned a lot about the coral systems here. And we can open it up for any questions anyone has. You can, now would be the time to unmute yourself as opposed to before, or you can type the question in the chat as well.
Yeah, question, comments, observations, anything is welcome, it's just don't be afraid to ask. Because I don't have all the answers.
Yes, so I have a question here. Nice talk, Matthias. I wanted to ask you that in your part two, when you described that there is a lot of variation in photosynthesis because of the effect of temperature. And in your subsequent part, you have the effect of photosynthesis or the effect of light. So I don't think that both of these selection pressures are actually going hand in hand. I feel they are actually antagonistic. So if you actually say that light and rhodopsins are actually creating a lot of variation, and that is what the species, the corals respond to more strongly, correspond to temperature. So the very point of bringing that climate change raises temperature in the depths of the sea might cause this variation could fall flat is what is my opinion, because your later part of your work actually shows that they are more light sensitive compared to temperature sensitive. And yeah, so I just want to know your opinion on that. And secondly is, when you talk about GPCRs, did you actually get any genes which are known to be related to thermal tolerance in your genomic data? Yeah, thanks.
Yeah, thank you for your input. So yeah, I didn't. If I came across as showing that the two things are related to speciation, like light and temperature, that's not something that I meant to do, because what I found in terms of bleaching susceptibility is different from the evolutionary analysis that I conducted in terms of looking for regions that are involved in a speciation or potentially in a speciation. So the fact that, as you said, if I were to make that conclusion, that could fall flat, that's actually true, because I'm not saying that speciation, temperature, and light are going hand in hand. What I found is that certain cues based on what we know in terms of reproductive biology and light photobiology of corals could actually be related to how they segregate and how they are subjected to different environmental or evolutionary pressures, right? And so I hope I have given you not my answer, but like my overall view to your comment. And for the genomic analysis in terms of temperature, we found one gene that is related to protein shock, heat shock proteins, but we didn't look further into that. So those two aspects were kind of a little bit not necessarily probably very well connected. It was something that as I was bumping into my research, I started kind of branching out and finding this pattern. So it would be great to actually test all this through either biological experiments, like controlling light, temperature altogether, but also probably using like gene knockout to turn or off or on those GPCRs genes. So yeah, that's my overall view of what you said.
Yes, thanks.
Thank you.
I had some questions, but just one simple one. Is the Media Luna, that's the site in Puerto Rico, right?
Yeah.
Is that special in the light penetrance as you go down to the depth? Like it is the pattern at that site special, or is that typical of most sites, or was that site selected because of the change of light through the
So
depth?
I guess the overall answer is yes to some degree, because depending on particular coral reef conditions, you can find that areas that are subjected to a lot of runoff or more are murky waters. Sometimes you see like the light the light penetrance and the distribution of species can be more compressed or more spread depending on how much light. So when you sometimes have corals that are closer to the shore, and depending on the particular bioptic, like the optic quality of the water, you can have like very clear waters, or you can have like murkier waters. And for this one, it is particular in the sense that we know that that happens in terms of light, but there are also some other studies from my advisor in another coral species, a soft coral, that they do segregate across the depth line. So for us, was like a great opportunity to actually go and test or look for genomic variation, because we know that that light penetrance is like one of the major factors that changes in just such a narrow depth gradient.
I see. Thank you. And we have a question in the chat from Andrea. She says, awesome talk. I think she was featured on the acknowledgments also. Could you remind me of the morphological variations or differences you measured between and within coral species? And how do you quantify these differences in the cryptic species?
Yeah, hi, Andrea. Thanks for being here. So for this part of my research, we only look at the number of polyps per unit area. So that's polyp density, how many polyps you found, like I think it was like around 1.5 centimeters. So we only counted how many polyps were found. And then based on the depth at which each colony was collected, and then compounded, or also looking at the lineage that we found they belong to, we just tested for differences in terms of the difference or the number of polyps across lineages, across depth, and across the combination of depth and lineage. So it was a very gross measure of morphological variability, because we know that within the calyx there is more variability, and the actual calyx can be wider, can be more narrow, depending on those light conditions. That's something that I would like to explore further. You're welcome.
And I guess one other question I had, unless there's a question from anyone else, is were those the SNPs, were they adjacent to the GPRs or GPCRs, or were any inside of the GPCRs? Sometimes you can predict if that will be a functional site or change some wavelength absorption.
Right. So what I did was, because we have GBS data, I look downstream and upstream 10,000 bases from the location of the actual SNP, and from there I conducted gene annotation. What genes are within that particular window, and some of them were rhodopsin-like, but many others were associated to calcium, cellular calcium, and we know that particular metabolic pathway, it is associated through to GPCRs. So we didn't pinpoint like a particular opsin-like light sensitive, but the fact that we know that even if you inhibit calcium metabolism prior to spawning, we know that we can actually inhibit spawning. So, the fact that we know from reproductive biology, observational as well as experimental biology, that calcium metabolism, and I found a lot of signals related to calcium that are very close related to the metabolic pathway of signaling in terms of GPCRs. But I didn't have like the golden gene that is the GPCR, and that's the actual amino acid that is changing how the wavelength is. I would love to, but that's still early stages for this kind of research.
No, the research is really great and comprehensive, even as it is now. And I had some hopeful thinking there. Maybe there was something else.
Yeah.
But thanks so much, Matthias, for this great talk. Really enjoyed it. Tons of data. Really appreciate you sharing your work with us. And we will return next week for a Greg seminar. And I just want to thank everybody for being here with us. Thank you, Matthias.
Thank you. Thanks, everyone. Bye-bye.